Good afternoon, everybody. Myself, Godali Chetan, working as an assistant professor in PG Department of Genetics, Walchand Center for Biotechnology, Walchand College of Arts and Science, Solapur. Today we are going to discuss about the laboratory design for animal tissue culture. The learning outcome of today's lecture is the students are able to explain and learn how to establish the animal tissue culture laboratory, and they are able to explain the animal tissue culture laboratory design. The introductory part and the laboratory design we are going to discuss in today's explanation of today's lecture. The culturing of the animal cells in artificial condition or in vitro condition is known as animal tissue culture. Harrison in 1907 actually started or grow the nerve fiber cells of the frog embryo first time to start the growth of animal cells in artificial condition. Later on, Carroll in 1912 is able to grow the connective tissue from the frog embryo and chick embryo for 2 to 3 months, and this success to the growth of the connective tissues of the chick embryo leads to the development of the animal tissue culture, where in 1920s and 30s the trypsinization method is developed, where the cells are separated from the tissue and these separated cells are used for the culturing purpose. Later on, it is observed that the number of different types of microorganisms are also able to grow and they affect the growth of the tissue culture. In 1940s, use of antibiotics into the growth of the tissue culture is carried out, and to prevent the contamination we use the different types of antibiotics. Later on, till 1950, the animal cells are used. From 1952, human cells can be grown in artificial condition. In 1980s, the DNA transfer techniques are developed, and these DNA transfer techniques are also used to transfer the foreign gene into the animal cells, and these animal cells later on grown into the artificial condition, so that we are able to know about the specific character is introduced into that particular cell or not by using animal tissue culture. After 2000, the human genome project can be started, and by using this project, initially animal cells can grow and the DNA from that cells can be extracted, and this extracted DNA is used to find out the number of genes present into the human genome as well as their functioning. So any one person is wanting to do the animal tissue culture and learn or to establish the industry regarding the animal tissue culture and perform all these things, then he or she must be able to know about the designing of the animal tissue culture laboratory, and for that purpose we have to discuss about the various points to be taken into the consideration when we are going to design the animal tissue culture laboratory. First part is the planning. When you have to plan to establish your own industry for animal tissue culture, then you discuss about the following points. First one is the ventilation. When you consider about the ventilation, the pressure present inside the laboratory is the first point to be taken into consideration. That always, if we are going to use the animal cells, then positive pressure inside the laboratory is maintained, and when we are going to use the human cell, then negative pressure should be maintained. Then second point is the accommodation. If you are going to establish the animal tissue culture laboratory, then how many people are going to work in your laboratory, that should be fixed. If simple and small scale laboratory is there, then not more than 50% can be work into that laboratory, and they cannot be working simultaneously. They cannot work in simultaneously. They can work in shifts. Then how they can prepare, how many different types of area are present, and which type of work they can carry out in this particular laboratory. According to that, we can provide the space for them. That number of different types of instruments are used. For that we require the space, and according to the design and the scaling we provide the space for the instrument as well as the working area for the number of different workers. That point, if we consider only when we are going to establish a new laboratory. If you have the already existing building and you have to make the renovation in this building, then there are limitations for that, because you are not able to make too much changes if your building is already exist. Then access, the number of peoples working in this area should able to access the laboratory and be able to access the various areas such as preparation area, washing area, sterilization area, hot room, etc. Quarantine is the part where the separate room is provided for them, and it is also taken into the consideration during the planning when you are going to design the animal tissue culture laboratory. The second part in this is the actual constructions and services provided during the animal tissue culture laboratory is the location of the animal tissue culture laboratory. It is not too much away from the city and it is not within the city. It is a little bit away, not too much away from the city, and it is located isolated area so that traffic is not too much, so as to avoid the contamination from the microorganism. Whenever you are establishing the animal tissue culture laboratory, the supply of filtered and sterilized air is important. Otherwise, it creates the anomalies during the animal tissue culture laboratory. The cleaning of glassware, plasticwares and instrument is carried out in routine basis in animal tissue culture laboratory, so that the cleaning area or easy cleaning is maintained within the laboratory, and whichever the dirt material and the waste material produced after the animal tissue culture, it is easily drained out from the laboratory so that it cannot affect the health of the worker as well as it cannot affect the normal growth and development of the animal cells. Then the number of workers and the number of laminar air flow fluids which are maintained into the laboratory they are placed away from each other. Approximately 2 feet distance is present between them, and head to head 8 feet distance is there, so that traffic should be avoided. Number of workers are not gathered simultaneously in this sterile working area where laminar air flow fluids are placed, so that traffic is minimum. And most important part in the construction and services is the electricity and the gaseous supply. Most widely the carbon dioxide is provided for the growth of the animal cells. All the instruments which are used in animal tissue culture laboratory, they require the electricity, continuous electric supply. So it is necessary when we are constructing the laboratory that we maintain the continuous supply of electricity as well as the CO2 gas. And vacuum line is also maintained so that the dirt material is supplied to the washing area. Before that, it can be passed from the vacuum so that it can be separated from that material, and it is easy for the cleaning purpose. Then we shift towards the actual layout of the aseptic room where sterile handling area is maintained. This particular sterile handling area is away from the other areas such as your preparation room, sterilization room and the storage room, and total aseptic condition is maintained in this particular room. This area is restricted for the workers only and not too much traffic is present in this area. Sterile air is passed in this area. In this particular area, laminar air flow is maintained, where class 1 type of laminar air flow is maintained outside, which are used for the actual working of the animal cells in aseptic condition. The laminar air flow where aseptic air is passed from the filter and positive pressure is maintained so that the entry of the microorganism from the other, if any case they are entered into the laboratory of this area, it can be prevented. The other room is present which known as quarantine and containment, where class 2 type of laminar air flow are maintained, which are used for the maintaining, culturing and studying of new cell lines, animal cell line, and the biopsies which are introduced into the laboratory before they are working before the peoples or the workers which are working in this laboratory actually start their research on that particular new cell line. Before that, they are placed, studied and identified for the any contamination in this particular area. Apart from that, service bins is also provided where different types of microscopes, cell counter are placed for the routine examination of the growth of the animal cells. This is the schematic representation of the simple animal tissue culture laboratory. You can see the various parts: washing area, laminar airflow, preparation area, etc., etc. Then, after culturing or transferring the animal cells into the vessels, they can be incubated in two different ways. One is the incubator and second one is the hot room. If number of cultures used and the vessel used are too much, then entire room is converted into hot room. This room is not specifically designed for the culturing of the animal cells, where it is insulated so that cold spots cannot be developed in that particular room. Where incubators are used when the number of cultures are less in number, and these incubators are used to maintain the proper temperature for the growth of the animal cells. After incubation, when we place the culture for the incubation, we shift towards the another part of the laboratory where preparation area is present. In this preparation area, three different parts are present, where one part is media preparation, second part is the wash-up, and third part is the storage. In media preparation area, we have to prepare the artificial synthetic medium which is used for the growth of the animal cells. In this, it is easy for us if a synthetic media is ready-made available. If it is available, commercially available media is used. If it is not used or it is not available commercially, then you have to prepare the stock of the media sufficient that the complete course of animal tissue culture can be carried out by using that particular media. Second part present in this area is the wash-up area, where the actual washing of the glassware is carried out, and the third part present is the storage, where different types of glassware, plastic wares, forceps, mask, hand gloves, lab coat, etc., and the other instruments which are used in the animal tissue culture laboratory are stored, and this area is near to this media preparation, wash-up and the actual working area, so that the worker is able to access this material which is immediately required for the culturing and carried out culturing of the animal cells. After the preparation area, we shifted to the reference, that this is the reference which is used, the Ian Freshney culture of the animal cells. Thank you for the listening.